Previously, we have seen how we can convert the raw data into the normalized data. But sometimes it is useful to obtain the raw data and to see the values of the raw data also. And after obtaining the raw data, we can compare the raw data with the normalized data, and we can see the difference, and we can also see the change in the data that helps in the quality control process. In case of FE matrix data, there is no such term as raw data because we cannot see the values of the data that is obtained after FE matrix experiments that is present in the probe sets. So we cannot see the values of the raw data in case of FE matrix. But in case of Agilent, we can retrieve the raw data by using the argument, that is, we can use the argument none when we using the process of normalization. And in the Lima package, there is a command that is known as normalize within arrays, and we can use the argument none and we can get the raw data in case of Agilent. Similarly, in the case of Illumina, we can also get the raw data by using the same process of normalization, and we can use the argument either using the term Lois or any other quantile in place of Lois or quantile, because they are the two different modes of normalization. Instead, we can use the term none and we can obtain the raw data, and this raw data is stored in an object, and we can then retrieve that object to see the raw data both in case of Agilent and Illumina data. Then after retrieving the raw data, we can store or save this raw data in some package of the bioconductor, and in this case we can use FE or bead array packages to store the raw data. And because most of the commands that are present in the bioconductor, they cannot read the data that is in the raw form, so it is better to use the data that is present in the form of tab delimited text file. And these text files are generated by just adding or pressing the button of tab, so that all the values, they are separated with the help of the tab. And this file can be read easily by the commands that are present in the bioconductor. For FE matrix and Illumina data, the expression values that we have already seen is transformed into log2 transformed, can be extracted using the command exprs, and within brackets we have to add the data. And we have to normalize this data, because we know that if we use the command dat.m, it means we are going to normalize the data. We use this command, and in addition, Illumina data should be log2 transform, because we have already seen that in case of Illumina data, we have to use the third step after background correction and normalization. We have to convert data into log2 transform data. For this, we use this command, and for Agilent data, the procedure is different depending on the data type. For two-color data, we should first extract the expression values and then assigning the genes their correct names. So we can use these commands datm and row names, and we can assign the names, and we can retrieve the names of the probes as well.